போட்டு அப்படியே பேசலாமா? உங்களுக்கு கேட்கும்ல.
இப்போ மியூட்ல இப்போ மியூட் பண்ணிக்கலாம்.
Hello.
Sir.
Pavan. Pavan audible?
Sir, your voice is audible, sir.
Okay, fine. Okay.
Sir, call you, sir.
Just to. Busy Pavan.
No, sir. Okay.
Busy. Okay, he's talking with somebody. That's why he left.
Actually, he came, sir. By 9:45 itself, he joined.
After that, I think he left because I think he's busy with something. That's why.
Wait.
சார் என் கூட தான் இருக்காங்கம்மா, நான் சொல்லிடுறேன் மேம். சார் மீட்டிங்கில் இருக்காங்க, நான் சொல்லிடுறேன். ஓகே.
Ramlingam, sir? Ramlingam, sir?
Just a second. Hello? Yes, sir. Good morning.
Hello? Sir?
Sir,
good morning, sir. Ramlingam.
Good morning, sir. Can you hear me?
Good morning, sir. Sir, welcome, sir.
Thank you, sir. Thank you.
Yes, sir. We can hear you, sir.
Okay.
You can hear us, no? No problem.
Pavan, you can start, Pavan.
Yes. Dear participants, today we have very special speaker with us. He is from National Institute of Research for Tuberculosis, Chennai. And before his presentation, I request Ms. Likita to give a brief introduction about him. Likita, can you please take over?
Yes, sir. So good morning to everyone for dignitaries here. Today I would like to introduce about Dr. V.N. Azagar Dustekar. He was working as a scientist in National Institute for Research in Tuberculosis, Chennai, India. Researcher in mycobacteriology from 1998 with MS PhD degree in microbiology in University of Madras. He has 20 years of research experience in tuberculosis bacteriology. He developed 3 genetically engineered bacteriophage consistents for diagnosing tuberculosis. He submitted a patent application for developing rapid method in diagnosing drug resistance in collaboration with IIT Kharagpur. He authorized 36 publications and contributed 2 book chapters. He was invited as reviewer for Asian Pacific Journal, found in tropical diseases, and Journal for Microbiology. He has active collaboration with IIT Kharagpur and IIT Varanasi, IIT DM, and IICT in CSIR and MKU. Apart from this, he is also having a following awards which he has received. Graduate researcher position in Albert Einstein College.
Ligita?
Graduate research is
Yeah, please continue.
graduate research position in Albert Einstein College of Medicine in New York for a period of 9 months. He also received Dr. C. Srinivasa Rao Award for Best Paper Award in tuberculosis by Young Scientist. He also awarded as ICMR International Fellowship to work in Loughborough University, UK for a period of 3 months. He also awarded a 2 lakhs rupees for developing the property for the proposed design entitled with microfluidics in development of rapid TB diagnosis under ICMR, IIT Kharagpur and MedTech program. So by this, I would like to invite Dr. V.N. Asgar sir to please deliver his words.
Sir, good morning, sir. Sir, you are on mute, sir. Please unmute, sir. And I have given you co-host privileges. Now you can share your screen from there itself.
Okay. Thanks. On the outset, very good morning to one and all. And I'm very glad to be here today among you to deliver this lecture about phage therapy. And before I begin, I would like to thank the organizers, Dr. Ramlingam and his team, excellent team, who worked for all these 6 days and making me a part of this 6-day program. So to begin, I belong to National Institute for Research in Tuberculosis. Just before the presentation, it's customary for us to tell a few things about my host institution, and then I'll begin my talk. Yeah. So my parent institution is under the headquarters of Indian Council for Medical Research, and now it is in the forefront in fighting COVID and helping out and diagnosing and treating the patients for COVID. So our center is one of the 26 permanent institutes under ICMR. And two of the major initiatives under this ICMR is India TB Research Consortium, which actually supports various TB activities and for clinical trials, new regimens, new diagnostics, and so on. And so the other main initiative about ICMR related to TB is TB Diagnostic Initiative. Under this, our center was able to now test and validate and made it recommended by WHO for diagnosing TB rapidly. It's one of the first indigenous cost-effective rapid molecular diagnostic kit for TB. It's called TruNat-Ref. And so this was now approved by WHO. This both was under the auspices of this India TB Research Consortium of ICMR. These are the two major initiatives among the other initiatives, including COVID, for being conducted by our headquarters ICMR. Coming to my host institution, National Institute for Research TB, there are a lot of contribution for TB management has gone into the program. And this has actually our findings and the publications have revolutionized the TB treatment chemotherapy. And this has told the world that home-based treatment is sufficient for treating TB patients rather than being isolated in a sanatorium. And it's our center was conducted one of the largest BCG vaccine trial in Chengalpet trial. Apart from this BCG clinical trial, around 50 randomized clinical trials have been so far conducted by our center. This BCG trial was one of the unique one, which was telling the world that the BCG vaccination for TB, which is given in India, is not giving that much protection for a adult human being. And this has led the world to squander and break the heads for bringing out a new TB vaccine. And our center has provided an evidence-based basic for the globally used directly observed treatment short course DOT strategy for demonstrating the role of supervision and documentation of TB treatment. Apart from this, we work in estimating the TB burden for the entire country, and we have conducted 2 phase HIV preventive vaccine trials, and so on. These are the few important initiatives and contributions done by our center. And coming to my department, where I am working in this in ART, I belong to a department called Bacteriology, Department of Bacteriology. I am basically a microbiologist. I am into working with microbacteriophages initially, and now till date we were using this microbacteriophages for rapidly diagnosing TB. And now we are we are proceeding for considering this microbacteriophages for TB phage therapy also. I'll be talking on those initiatives also in the due course of my presentation. And apart from that, we are into developing innovative and rapid diagnosis for diagnosing TB at an earlier stage. Apart from this, the main work I work with is a factor called resuscitation promoting factor, and exploring novel delivery vehicles to increase efficacy and reduce the toxicity. And also we do indulge ourselves in discovery of newer anti-TB molecules from various sources and ecosystems we receive from other centers out from India for screening them against TB. So these are the major activities I am personally involved. So coming to this phage therapy, before I enter into this topic, I would just like to brief about bacteriophages. So basically, these bacteriophages are viruses which have been now talked throughout the world by the illiterates also. People are now more aware of what is a virus now. So to just give a glimpse of what is bacteriophage, how it is differing from the other viruses. Actually, a bacteriophage is a virus which can infect a bacteria. So it makes bacteria, gains entry into the bacterial cells, cytoplasm, and goes inside and replicates and forms newer virus particles. And then it breaks open the bacterial cell and comes out and again infects a newer bacterial cell. So this is the concept of a bacteriophage. And so the phage therapy utilizes this bacteriophage, this adorable phenomena of this bacteriophage that could specifically target and kill the bacterial pathogen. Just to begin with this talk with a history of bacteriophages so that you will understand where bacteriophage began. It actually began before the era of antibiotics. So now I think the pharmaceutical industries will have a very prompt role in manufacturing these bacteriophages and going for formulations of these bacteriophages. And so definitely you'll be playing a very active role in this form of therapy, newer form of therapy in the coming near future due to the advent of antibiotic resistance. So coming to bacteriophages, phage therapy actually, use of bacteriophages actually started before around 18th century itself. From 1896 onwards, bacteriophages have come into fore, and it has been recognized by various researchers. I'll be talking in the next slide about these researchers and their contributions and due course. It was actually isolated in pure form in 1920, and later in 1930, it was licensed for phage therapy. It is being proposed as an alternative to antibiotics due to the emergence of multi-drug resistance. And first trial was done in 1931 for treating cholera. Well, it happened in India itself, in Punjab. Later, the phage preparations of Staphylococcus, Streptococcus, E. coli, and other bacterial pathogens were prepared by several entrepreneurs in Brazil, US, Poland, Georgia, and so on. So main advantages include safety, specificity, and capacity to deal with the biofilms also. So just to talk about the broad history where it all began related to this use of these phages for phage therapy. In 1896, Hankin actually termed it as an antiseptic and called that he found that it is having an antiseptic action of river against Vibrio cholera. So he found unique microorganisms which was playing a vital role in clearing out this Vibrio cholera in river water. So later, Malia described it as that it can ferment, that can destroy bacteria in 1898. Later on, in 1915, Thwart et al. And this group actually termed that they are bacteriolytic agents of enzymatic nature. So, but T. was the person who gave the name of these unique microorganisms as bacteriophages and defined that bacteria are susceptible to infection that are host to ultramicroscopic agents, and he termed them as bacteriophages. It was in 1917. Later, 2 years later, the birth of phage therapy occurred, and here they use these phages for curing cases of dysentery using these Phage blocks. So later on, it was expanded to dysentery, bubonic plague, conjunctivitis, skin infections in 1920, and subsequently in another 9 years, Alexander Fleming actually accidentally end up in noticing the action of penicillin notatum against the bacterial pathogens. He accidentally discovered this group of fungi which has shown a remarkable activity against the bacterial pathogens, where the antibiotic era actually began from 1929 onwards. But during 1933 and 34, the researchers in bacteriophages actually made a critical review about this phage therapy and put forth what are these trials, what was the lacking features in these trials, whatever had been conducted till date, till 1933. They found that it lacked controls. They found that there was very low efficacy of these phages, and the possible co-founders were like low purity of the phage lysates. This actually made a setback in the phage therapy trials. Till then, the exact phage morphology, phage life cycle was not deciphered. So before that, they actually thought of using for curing infections. But before finding out the exact life cycle, exact phage preparation modalities were standardized, so they end up in a little bit failure. Setback was there. So what are the causes? They actually put forth these things that are controls where the efficacy was very low purity of the phage lysates during 1933 and 34. So later on, at 1937 and 39, Ellis and Delbrook actually characterized the various features of phage life cycle, wherein the parts of each and everything in the life cycle, phage life cycle was deciphered, and they put forth what is absorption. It goes the bacteriophage. How does it get attached with the bacterial cell? They call it as adsorption. And what happens inside the bacterial cell? It actually attached to the cell surface. It injects its bacterial DNA into the cytoplasm of a bacteria, and there on this the DNA which enters the cell wall of the bacteria, there it replicates and produces multiple copies of the phage particle, and then it actually lysos open and comes out. This entire life cycle was deciphered by Ellis and Delbrook during 1937 and 39. So later on, its first electromagnetic microscopic image was documented by Raska in 1940. But after 2 years later, penicillin came into forth and took on against versus this bacteriophages. Till then, bacteriophages could not do wonders. So penicillin, they found that it could do wonders, and it was very much useful for curing and saving the patients and the soldiers during the World War time. So that began the antibiotic era. So the golden age of antimicrobials took on later after 1940 till 1970. Over around 40 antibacterial compounds, antibiotics are discovered and introduced, namely beta-lactams, aminoglycosides, tetracyclines, and so on. So the golden age of antimicrobials took over and completely masked the field of bacteriophage during this period. But around 1977, the first, but there was a little bit of phage research was going on behind and in Russia and certain countries in Russia and Poland, so on. But a little bit in Western and European countries also, they were into sequencing, and the first phage genome was sequenced in 1977. But forefront antibiotics was very much flourishing during this period. So in 1983 and 1986, Slopak, Smith, and Huggins actually indulged in phage therapy against antibiotic-resistant bacterial strains. So from this period, 1980 onwards, people came to notice the drawbacks of antibiotics, wherein the bacteria they began to develop resistance. And so people were thinking, what will be the next alternative? So the major group actually went in to address the bacterial resistant mechanisms and to combat this resistance with the newer antibiotics, newer analogs of antibiotics, so on. But the other group actually felt the necessity that chemical antibiotics want to a good thing on a long run due to the emergence of bacterial resistant, antibiotic resistant bacterial strains. And so Slopak and Huggins actually went into phage therapy against this antibiotic resistant bacterial strains in humans and multiple animal models. Later on in 1992 and 96, Sutil Merrill et al. Actually went in for phage therapy studies in animal models. A number of new antibiotics started decreasing during this period around the millennium. So they usually term it as dry pipeline because of the adverse side effects which was noted for by the use of rampant use of these antibiotics. So there actually the regulators and the health authorities from various countries set into forth a lot of regulations for this antibiotics use of antibiotics for humans. So lot of ethical issues and regulatory issues came into foray, and so the issue of newer antibiotic coming into market was much more very costlier affair during this millennium and now also. So to handle this regulatory issues and ethical issues, people are struggling to bring out newer antibiotics in the forefront. So this era was termed as dry pipeline. So not much of a newer antibiotics could be seen after 2000. Uh, but phage therapy later on slightly began in 2006. Around first commercial phage-based biocontrol product was launched against this Listeria monocytogenes, and 2009, Wright et al. Did indulge into the first phase trial of first and second phase trial, controlled clinical trial using this phage therapy in chronic otitis, which was caused due to multi-drug resistant Pseudomonas aeruginosa. And later on, multi-centric studies are using in the phase 1 and 2 trials were also conducted subsequently. So this is the broad outline began, and now what is at the stage we are. So still, lot of things has to be addressed as we indulge into clinical trials, and a lot of issues have come up in the phase therapy also. So I'll be talking into the subsequent slides about what are the challenges also before summing up about the bacteriophages. So this slide actually depicts the advantages about bacteriophages versus the antibiotics. Antibiotics are, as you know, that they not specifically targets the normal microflora apart from the bacterial pathogen also. But bacteriophages are very, very specific. A group of bacteriophage can infect that specific host only. It never cross reacts and cross infects the other bacteria. So a bacteriophage, if it has been characterized to be isolated from a host, it infects that family of bacteria alone. So that is the main advantage of the use of these bacteriophages, which are very much specific enough, and antibiotics are being metabolized and eliminated from the body in due course of time and may not be available at the site of infection. So you need to have repeated multiple doses of antibiotics in a close duration. As a short frequency. You need to keep on giving the dose of antibiotics until you get the eliminating the pathogen from the human body. But unlike antibiotics, bacteriophages can replicate at the site of infection. Actually, bacteriophages, for that matter, any viruses, if it is going to be outside the living cell, it's called as a bacterial particle. They are non-living when it is outside the cell. It requires a live cell to get itself multiply. If you make the bacteriophages to reach the site of infection, it goes inside the bacterial cell pathogen and multiplies and produces new phage particles. This slice open the bacteria, kills the bacteria, and comes out and again infects the other bacteria which is near the vicinity, the site of infection, and goes on replicating and so on. So you need not give multiple doses of bacteriophages. One shot of bacteriophage should keep on replicating, and so it increases the bioavailability until the host cells are there, other the pathogen are still there in the site of infection. Whereas antibiotics has multiple severe adverse reactions are met with the use of antibiotics, but till date, from the clinical trials and animal model trials, bacteriophages, not much of serious side effects were noted. Whereas antibiotics, newer antibiotic developing the newer antibiotics is very, very time consuming, and it's very cost inducing factor. It requires a lot of money to bring out a newer antibiotic into a market. But bacteriophages, selecting newer phages upon the emergence of phage resistance is also very, very relatively rapid. And so you don't require that much of infrastructure to identify newer phages also, apart from propagating the newer, the older phages. So these are the main advantages of bacteriophages, which stands against antibiotics. So how basic, I'll just give an idea how these bacteriophages are handled in the laboratory and how they are multiplied and grown in the laboratory. So for this, you need to have that particular host of bacteria which is required for infecting this, which is required for propagating this virus. So when you end up in isolating a virus, you know the host which it actually infects and produces multiple viruses. So after identifying the virus, you should use that host bacteria. You can use that same host bacteria to culture them and propagate them. So you need to have the bacterial cultures. You add the virus to that one, go for incubating at an ideal temperature, and then you can take multiple copies of the viruses would have been produced, multiplied inside the bacterial cell, and then you can filter out the bacterial cell and filter out the virus particles separately, and you can have the phage lysate in your hand. So when you want to test whether how much of bacteria viral cells are there, you can just spot it out on lawn of bacterial cell. So you have to grow the bacterial cell in a petri dish and then spot your virus lysate, the virus lysate over the lawn of the bacterial cell. And after further incubation, you can look for the plaques. As you see in the top right corner, the plaques represent the phage particles inside the empty spaces. The other the turbid ones are the bacterial cells, uninfected bacterial cells. So these are called as plaques. Under this, each plaque you'll have hundreds of viral particles. So from this lawn of phages, lawn of cells, you can remove the viral particles. You can filter out the bacterial cell and take the phage lysate, and you can go for harvesting them and multiplying them. So this is how phage is being propagated in a lab. This comparatively it doesn't require a much more sophisticated infrastructure. Basic laboratory is enough for doing this fast propagation and multiplication. So initially, in the phages are being grown, multiplied, propagated. Here, instead of culturing, we use the term called propagation because you require a live cell to grow them. So that's why it is called as a propagation. So this viral multiplication was will be usually adapted using the use of the solid media. But if you want to have a huge amount of these phage lyses to be prepared, you can go for liquid culture propagation also. So that now people are working in optimizing large-scale production of this liquid culture phages, and biofermenters are also there in the market, which will be used for propagating this virus large scale. And so that is how the phage can enter into the market for large-scale preparation. So what are the commercial values of these phages? Actually, De Herel in Paris actually produces five phage preparations, and now this company is termed as L'Oreal entered into cosmetics. In 1940s, Eli Lilly company produces 7 phage products for human use, targeting staphylococci, streptococci, Escherichia coli, and other bacterial pathogens. Mainly Georgia and Poland, there are two big institutions, Elevia Institute of Bacteriophage and the Esfil Institute of Immunology at Poland. are currently involved in phage therapy, whereas in India, Vitalis Phage Therapy, the company is into phage therapy. They actually target the disease prostatitis and epididymis. So, what are the possible routes by which you can make the bacteria, the phages, to be administered? Oral, rectal. For oral, it is you can make it in tablet form, and the liquid formulations are also there. You need to have around phages at the dose of 10 to the power of 11 PFU. It is called block forming units per dose. So you need to have that kind of tighter strength of the phages per dose, so you can use this at this concentration for oral delivery, rectal, local administration for through skin, ear, and eye, nasal mucosa. It can be applied in tampons, rinses, and in creams. These are the formulating forms by which you can go for local administration. Intranasal delivery for respiratory pathogens, intrapleural injections, and intravenous venous also you can use the phages, and they have been used in different cases also. I will be talking about another case study in the last few slides. So now, what are the main challenges in phage therapy? So after understanding phages, understanding its specificity, so when you want to end up in formulating them, when you want to do it in a larger scale, and when you want to use it for human trials, what are the major challenges? So these are all been populated here in the slide, and these are all been populated due to the various other clinical trials or other difficulties met in the various clinical trials and animal trials. So based on their observations, we have populated the main challenges. So you need to have proper purification of these phages, and its storage temperature, the safety and efficacy are all depends upon the proper purification of these phages and its relevant storage. But now there are ample number of ways by which you can purify these phages. Little, very small filtering units are also available now to purify the phages from the host debris. And what are the storage conditions? This is another thing we need to sanitize before it enters into the market and for industrial preparation. So its storage conditions are also for each and every bacteriophage, different types of bacteriophages. The storage conditions are being defined, and you want to end up in treating a specific disease. When you have the set of phages, you can go and do a study. You have to do a study related to the storage. Actually, what type of freeze drying or lyophilization is it requires, and what are the rehydrants which are required to bring back the phages in that particular dose. So these storage conditions and safety stability has to be studied for each and every phages being isolated, and you want to consider them for treating the bacteria. So delivery of the phages at the site of infection is also very, very important. You need to address it for every other specific disease you want to treat with these phages. So this you have to handle before you think of animal model or human trial. And the complications in designing the phage cocktails, never ever you can use a single phage for any disease treatment purpose. So you need to have multiple cocktails of phages for treating. So the need of prepare a phage cocktail is very challenging due to the narrow host range. So you need to have a proper cocktail, and cocktail, when you want to use it against a pathogen, you need to test them in the lab before you want to consider them for human trial. So the poor stability and viability of phages, they are very much unstable. So the viability are very much limited. So this thing you need to address. The lack of knowledge of its biology, heterogeneity, and the mode of action of each and every phages also are very much important things to be addressed earlier, before when you want to try them out for the curing the disease in human. So induction of neutralizing antibodies is also another thing you need to evaluate this development of antibodies against these phages when you want to consider human trials. So lack of a specific regulatory framework and legal issues related to IPT, Intellectual Property Act, is also very much of concern of late. So another thing is it's very technical. Lysis from without and lysis from within is also another property of phages. You need to address these things. It's very technical. I'll just leave out this lysis from without and within. So what are the different novel approaches of phage therapy? How can you administer? You can have a conventional phage therapy using monophage or a phage cocktail. Mostly the phage cocktail is the advisable one. You can have a immunophage antibiotic combination also. You can have the immune bodies tagged with the phages, or you can have the antibiotics to tag with the phages and go for a combination of therapy. Phage derived proteins, actually the phages, when it enters into the bacterial cell, it secretes an enzyme called lysine. Due to the effect of this lysine is the main culprit which can lyse open the bacterial cell and come out. So this lysine also can be considered to treat the bacterial pathogens. So the phage derived protein also can serve and lead us to phage therapy, whereas the bioengineered phages also can on a long run can be thought of going for a phage therapy approach. This also can be a novel approach, and people are working in bioengineering phages to attach the novel delivery mechanisms or novel molecules or targeted approach can be thought of using bioengineered phages. Phages can be used for bioengineering purpose. They can be they can accommodate foreign proteins also into the head of these phages, and so they can be bioengineered, and this bioengineered phages can be used for therapy. So these are the novel ventures where we can use think of phage therapy and make it much more efficacious. So what are the pathway for phage therapy research? You need to characterize, you need to isolate the phages. This is the main outline by which you can go to think of the phage therapy. First initial thing is you need to have a good pool of phages against that particular bacterial pathogen. You need to isolate them, have in large numbers a different type of bacteriophages against a particular bacterial pathogen. You should have methodology to isolate them and go for purifying the phages also. So after purifying and isolating the phages, go for the host range determination, whether it can infect the various other species belonging to the same family of bacteria or not. And you need to have the address, you need to find out the multiplicity of infection. That means how many phage particle can accommodate, can be accommodated by a bacterial cell. And at that stage, when it can open the bacterial cell, lies open and come out. So that is called as a multiplicity of infection, and it has been used in different term while in the lab also. So this MOI also has to be determined for this much number of bacterial cells, this much number of phages are required. So that ratio has to be is different from each and every different bacteriophages. So this MOI has to be uh, documented before that when you want to think of phage therapy. Then you have to characterize the shape, absorption rate, the latency period, the stability, and the burst size also. So later on, after having all these things in place, then you can think of large scale production, designing efficient delivery systems and clinical efficacy testing. So you need to have a proper system to test them. For example, for respiratory pathogens, they have a system called multi-stage liquid impactor. So this actually can represent a human lung. So this apparatus is used for delivering and used for testing whether the phages can enter into the lungs and can it reach the alveoli at that particular size. So using this lung model apparatus. So you need to have a basically you need to have a paka delivery system for respiratory pathogens. You can have the nebulizer, and for you need to go for the choice of nebulizer also. There are at least 3 nebulizers in the market now. Each and every nebulizers differs for delivering the phages at a right titer. So this nebulizer also has to be tested for each and every phase. For example, bacteriophage which can infect TB, it is called as D29, behaves well with the jet nebulizer rather than the other usual vibrating mesh nebulizer. So this nebulizer also actually, while it produces the droplets, it actually reduces the titer. Some kind of nebulizer reduces the titer while it delivers, generates the aerosol. So the nebulizers also has to be tested, and we need to uh, you need to arrive at the right kind of nebulizer and the right kind of clinical efficacy testing apparatus also before you want to think of the phage therapy in human trials or animal trials. So this is the pathway, actually, brief outline of the pathway for phages to be considered for the phage therapy. So this actually slides depicts lung model apparatus for which was used for testing the phage delivery mechanism in lieu of lung infections. So this you can see the nebulizer fitted with the nose piece. This is the Alberta throat representing the human throat, and this is the multi-stage liquid impactor. It has 4 compartments, and the final compartment at which you can expect the particle size to be at around 4 microns to reach at this particular compartment, the fourth compartment. So this whole apparatus is been engineered in one of the labs in UK. They use this one for testing out whether the phage can reach the lung or the alveoli at the proper titer or not. So from each of the compartment, you can remove the phage slice it after it has been nebulized, and after it crosses through the throat, alveolar throat, and it reaches each other in every compartment. Each and every compartment there is an outlet. You can remove the phage slice it and to check for the titer. So at the final compartment, the fourth compartment, the size of the particle is usually around 4 microns. So whether that phase, that aerosol droplet, whether it can reach the final compartment and find out the titer, how much the drop in the titer, the strength of the phage is there in the final, when it reaches the final compartment, can be elucidated by this lung model apparatus. So this is an example for which I gave you, I am presenting you is for a lung model. It is an in vitro lung model apparatus where you can see whether the phage titer is right or not when it reaches the throat or when it reaches the final compartment. So, so you need to have a foolproof in vitro clinical testing system to test whether the phage is reaching the desired site of infection at the proper dose. So apart from this, I just want to give the successful case study report of personalized medicine using phage therapy. There was a case, this was brought out by Schooley and his group in 2017, wherein a patient who was infected with a disseminated resistant, multi-drug resistant Acinetobacter baumannii infection came into the hospital, and he actually he was tried out upon different antibiotics in the world. None of these antibiotics could wipe out this Acinetobacter, so it was that much resistant, and later on the patient slipped into coma. So here the patient was declared that they didn't have any available options. So he was the ethical committee gave them the approval for treating them for any other novel therapeutic platforms available for treating this infection. The patient was in coma, and so he was actually he was around a 62-year-old diabetic patient with necrotizing pancreatitis with a multi-drug resistant Acinetobacter infection. So in spite of multiple courses of antibiotic courses, the patient deteriorated over a 4-month period, and later on he went into coma. So then the people was thinking of using bacteriophages, the last ditch effort, whether it can bring out the patient from this coma or not, whether to save the patient. They tried out using different bacteriophages. They actually administered these phages. Here actually they use Navy and company, Texas University, along with a commercial company. These all 3 people and collaborated and devised different mechanisms for administering phages. They had luckily they had a group of phages which can infect this particular pathogen. So using this multiple combination of bacteriophages, they brought a remarkable result. They administered them intravenously and percutaneously, and they found a reversal of the patient from the downward clinical tragedy. They actually ended up the treatment with this bacteriophages. Of course, they were combined with the last ditch effect of one of the antibiotics, which was sensitive. They used that antibiotics in combination with the cocktail of phages and cleared out this infection. The patient began administering these phages within 48 hours. The patient came out of coma, and completely another 200 days of treatment with this different cocktails of phages. The patient had cured from this infection completely, and now he's out to work. So this is one of the case study, but there are multiple case studies wherein they have used phages for successful bringing results. So here, the U.S. Navy used to combine multiple cocktail of phages, and Texas University they had a group of phages against this Snatobacter, and using all this cocktail of phages with different modality of route of administration. They first initially tried intracavitary, then went into intravenous, and further in the last 2 weeks, around 221 days, at that time period, they ended up in successful finding out the right combination along with the meropenem and antibiotic. With that, this combination intravenous data administration, they ended up in bringing back the patient from coma. And during this case study, they were able to find out the PK levels of these phages also. So they actually found out in that person, but basically in human, the ideal peak hours of the phage therapy remaining in this plasma was around in the order of 5 minutes only. In 5 minutes, it was in the ideal peak dosage in the human being, but at 30 minutes, it came down. So he required multiple injections, but earlier this was not thought. But now the phage PK also actually is required multiple doses. That was another result. This was a phage which was used for treating this patient. This is an microscopic image of this parts. This has a head, tail, and the fiber part which can actually get in touch contact with the bacteria. These are the different cocktails of phages used, and the ultramagnetic microscopic image. Discussed in this particular case study was that a clear turning point was observed within 48 hours of starting of intravenous administration of this phage, and the patient recovered as soon as it was started within 48 hours from coma, recovered from coma. Additive or synergistic activity between the bacteriophage and traditional antibiotic was demonstrated. Emergence of phage-resistant bacteria also was demonstrated, but it was avoided, and this delayed the combination therapy. Finally, with the addition of antibiotics and the right phage combo, the phage clearance was the ended up, and so it was on an order. The phage clearance in the system, human system, was on an order of hours. But it was when it was tested in rat model, but in this human patient it was in minutes. So when you want to use the animal trials, so you need to carefully think of the order of the phase clearance. So it will be in human trial, it will be in minutes only. So these are the important points to be discussed using this case study. So there are in the Googling out, you can find out numerous other case studies of use of phages, but still it has not ended up in the market because of ample availability of these antibiotics. So with this slow progress in the discovery of novel classes of traditional antimicrobial agents, novel approaches like phage therapy are much more needed to address the issue of antibiotic resistance. So with this slide, I end up my talk. I hope this will give you an idea about the phage therapy and their uses. Thank you. Thank you for patience.
Thank you, sir. Thank you for your very useful information and very knowledgeable sharing about this phage therapy. And so in the question answer session, we have few questions some of the participants have raised. I think that is visible to you also, sir. Question answer
Just a
session.
second. Question answers, is it visible?
Sir, below,
Yeah. So first question from Santosh Anasuri. Can we use this for TB and leprosy? Yes, definitely. There are animal trials are also going on for use of these bacteriophages. We call it as mycobacteriophages. There are a couple of people in UK trying out these phages for TB also. And leprosy also can be thought of, but testing mechanism is very much limited, because leprosy, the bacteria responsible for causing mycobacterium lepra, it cannot be grown in an artificial media. So that is another limiting factor for ending up in vitro studies with leprosy. But there is possibilities there. For TB, there are half a dozen mycobacteriophages in the world which can infect and wipe out this TB bacilli, and it has been documented in some certain animal models. So definitely it can be used for thought of using clearing TB and leprosy. So next question from the same person is there any commercial products in India? There are there was some company called Ganga Jain. No, it is not there, but they ended up in tropical applications for clearing wound infections for particular for each and every pathogen. So tropical applications are there, but not available frequently available everywhere. So next one is, can we combine with other antimicrobials? Yes, definitely. In this particular case study, I have discussed about using of antimicrobials. This has actually brought out a remarkable achievement, success when you want to combine antimicrobials as well as phages. In this particular case study, they met a complete success by use of tagging with other antibiotics only. In what physical forms these can be produced? Physical forms in powders, it can be produced, it can be lyophilized, it can be spray dried, or it can be freeze dried. So these are the various forms you can prepare the phages, but in each and every method of lyophilization preparations, you need to test the strength of the bacilli, the strength of the virus, and in each of the forms. For every bacteria, this the titer differs, its strength of the viral load differs from each of the every preparation modality. So, for example, one of the phage can be much more viable in phase freeze drying, but the same can be very poor in spray drying, lyophilization also for that matter. So, powdered form, but ultimately you need to reconstitute in the particular rehydrant, and the concentration of that rehydrant. Most people use trachelose. The concentration of the trachelose to be used also has to be tested out and um, used. So in liquid forms as well as powdered forms, the phages can be available. So what next? Next question is person Raghuvir, good a friend of mine. Yes, as human body also hosts millions of bacteria and other microbes. So will this phage therapies destroy the useful symbiotic bacteria? What is your take on this? Yes, actually, bacteriophages are much more specific in their nature. So it does not literally harm the other normal microflora. But the only thing is, among the same family, it can infect the other species. But the same family of bacteria microflora is not that much met in human being. So it will target the same family of viruses, bacteria, but other family does not infect. So it is very much specific for the family. Among the species itself, in the same family of bacteria, among the few species will not, it don't infect a few species, but other species. But they are very much specific for each and every family of bacteriophages. So literally, you need not worry about whether it can infect the microflora. That is the main advantage of using bacteriophages. So it is very specific in its targets. So these are the set of questions. And another question, lifespan of bacteriophage. So the bacteriophage lifespan depends upon until it has a host cell, live host cell is there. So the bus, it can be called as bus size also. So as soon as it enters the bacterial cell, it actually multiplies and produces multiple bacteriophages, and it lies open and come out of the bacteria in a specific period. So when it comes out of the bacteria, you need to have another live cell of bacteria to continue its life progeny. If there is no live cell outside the bacterial cell, then its life ends there itself. So it is in order of minutes and in order of hours for certain viruses. So this life cycle has to be found out. So that's how we call it as the burst size. So these are the few questions I saw here. Is there anything else? Any questions
No, sir.
from the
Dear
faculty?
participants, if there are any questions from YouTube viewers, please drop in the chat box. And those who are in the Zoom, please drop in the question answer session.
And
I think there are no more questions, sir.
Pavan?
Request Dr. Ramalingam to take over.
Yes.
Thank you, sir.
Sir, it's my pleasure to be in this platform and to see your and listen your talk on the factor of age and your therapy. And sir, really, it's valuable information, and it's the need of the hour even for this the growing infectious disease in the particular era, and especially if the drug resistance is also one of the major challenges ahead, where we are supposed to face with the things. And they can see that from 2000 itself, this phage therapy also introduced things, and the people now got some idea about phage therapy. And sir, with this, and personally as well as on behalf of RIPER faculty and students of the institution, and we would like to thank you for your valuable presentations, and thank you for your acceptance to be a part of this program. Thank you, sir. Thank you very much.
Sir?
Hello, Pavan.
Sir?
Sir left, I think, right?
No, sir, he's there, sir. He is under mute.
Okay, okay, okay.
Asgar, sir? Sir?
Sir.
Sir, he is under mute, sir.
Okay.
Sir came, sir.
Sir, thank you, sir. Thank you very much.
Thank you, sir.
And,
Thanks a lot, sir.
Thank you, sir. Thank you very much. And really, it's a pleasure to be with you and your presentations actually. And people are really, it's some kind of a new topic, and where the people come to know other than this synthetic molecule, chemical therapy, that therapy, this therapy is something new where people can have insight on this. And really, on behalf of management and personally as well as the faculty side, I thank you and your organization for your valuable presence and your presentation. And thank you very much for your being a part of this eFaculty program. Thank you, sir. Thank you very much. And your contributions we require always, and today, and not only today, forever. Thank you. Thank you very much, sir.
Definitely, sir. Definitely, sir. Thank you, sir.
Thank you.
And dear participants, afternoon session will be starting at 2:30. I will be posting the links in your WhatsApp groups by 2 o'clock, and please try to log in by 2:20. Afternoon session by 2:30 by Dr. Srinivas Mutalik, Professor and Head, Department of Pharmaceutics, Manipal College of Pharmaceutical Sciences. Thank you all.